So first of all, I would like to thank the organizers for inviting me to share the latest results of my lab with you. I was really hoping to be there in person, but unfortunately this was not possible. But I hope that I can share with you today a little bit about cellular senescence, skin and longevity, the strategies that we are using to study mechanisms of skin aging, and also to address these senescent cells in the skin. So I like to start my talks by advocating for the study of skin aging. There is the skin is our outermost organ of the body, and it is the organ that is in contact with the environment. So it's a protective organ. And during aging, the barrier is decreased, which means that we are more prone to get diseases and damages from the environment. Also, skin is a social organ, and skin aging can also have a negative impact on self-esteem, body image, and therefore in quality of life. So therefore, it is very important to study skin age. I think that at this point, you have heard enough that there are several hallmarks of aging, and among them, the accumulation and persistence of senescent cells in the tissues is one of these hallmarks. But we are not just interested in cellular senescence, but we are also interested in another hallmark of aging, which is mitochondrial dysfunction. And we still don't know, and there is a lot of debate in the field about if mitochondrial dysfunction is a cause or consequence of cellular senescence, but they are indeed interconnected. And it is also known that accumulation of senescent cells in the tissues is leading to age-related diseases, and that xenotherapeutics that are targeting senescent cells either by killing these senescent cells or by inhibiting the activity of the senescent cells have some kind of rejuvenating power. We and others were already able to demonstrate that in the skin, during aging, there is an accumulation of senescent cells, both in the epidermis, which is the upper layer of the skin, as well as in the dermis, which is the lower layer of the skin. And we know nowadays, using organoids, that by the time that we see senescent cells in the dermis of the skin, we have an aging phenotype in the epidermis of the skin. And this is changing based on the percentage of senescent cells that we have in one compartment, which means that there is a communication between the cells of this tissue. And then I would like to go back a little bit about what are senescent cells. I think this was already quite explored in this meeting, but I would like to highlight that senescent cells don't have a single type of phenotype. They have several characteristics that are not common to all of the senescent cells, but most of them, they have an increased, they have altered morphology, they are growth inhibited, but especially for our studies, they secrete a set of chemokines, cytokines, matrix metalloproteases, and nowadays we know that also extracellular vesicles that are consistently together call it the senescence-associated secretory phenotype. And senescent cells also have, most of the times, impaired mitochondrial function. The issue of these senescent cells in the tissue is that when they are removed and they have this transient SASP, this SASP is beneficial for the tissue. However, upon persistent SASP and persistent mitochondrial damage that we know nowadays that is also inducing the SASP, these cells, they can be pro-inflammatory and cause chronic inflammation, tumor progression, and age-related diseases. So these are the two main topics that we are interested in our lab: impaired mitochondrial function and the secretome of senescent cells. Based on this, we have developed a strategy in which we try first to understand molecular mechanisms of cellular senescence and mitochondrial dysfunction. Based on this, we find targets for intervention, and then we are also trying to find strategies about how to intervene on the skin. So in the next slides, I will share with you two targets that we have found based on molecular mechanisms, and I will discuss with you the strategies that we are trying to develop to intervene on the skin. The first target was based on a model that we call the UVB-induced senescence of human dermal fibroblasts. This model In this model, we irradiate human dermal fibroblasts with UVB twice a day for 4 consecutive days. And at the end of 2 weeks, we have a full senescence phenotype that is characterized by growth arrest, increased activity of senescence-associated beta-gal, and expression of senescence-associated proteins. So based on this model, we have started studying mitochondria physiology, and we have seen that UVB induces fragmentation of the mitochondria. And this fragmentation of the mitochondria is initial step for the elimination of this fragmented mitochondria by mitophagy. We have then nailed down the mitophagy receptors that would be acting on this elimination of damaged mitochondria, and we could find out that the mitophagy receptor NIX was not just upregulated by UVB, but also it changed its subcellular localization. So in control cells, the NIX was mainly localized in the nucleus, while in irradiated cells, NIX migrated to the mitochondria and primed this mitochondria to be eliminated by mitophagy. So there we found a target. Based on a molecular mechanism, we found a target for intervention. So when we do this, what happens is that we knock it down. So we transduced cells with lentivirus to deplete NIX in our cells, and when we irradiated our cells, our NIX depleted cells with UVB, we could discover that indeed NIX is a essential mechanism for the development of senescence, and in the absence of canonical mitophagy, the majority of the cells were not able to cope with the UVB stress, and they were not going to senescence. Additionally, these cells started secreting extracellular vesicles, and these vesicles contained damaged mitochondria, suggesting that this mechanism is an alternative mechanism of quality control to complement the absence of the canonical mitophagy. But simply independent of UVB, knockdown of NIX, so depletion of this mitophagy receptor that is responsible for the elimination of damaged mitochondria upon UVB, already leads to cellular senescence, to premature senescence in human dermal fibroblasts. And this premature senescence is also characterized by accumulation of damaged mitochondria and also by secretion of extracellular vesicles containing damaged mitochondria. So based on this target of intervention, we have developed our organoids, as it was explained by Miriam before. So these are very important tools for the study of physiology of tissues. And in our organoids, we seed the fibroblasts in a collagen matrix. And then on top of this fibroblasts, we seed the keratinocytes. And after these, we put these organoids in what we call air-liquid interface. This is ALI, and this means that the epidermis will be in contact with the air, while the dermis will be in contact with the medium, mimicking the conditions of the skin. And after 7 days, we have a fully developed skin model. So we have prepared, based on these mechanisms of intercellular communication, we have prepared organoids containing always wild-type keratinocytes and either wild-type fibroblasts or NIX knockdown fibroblasts. And before air-liquid interface, they look at more or less the same, but 24 hours after putting these organoids into air-liquid interface, we could already see macroscopic differences in these tissues. And when we analyzed the histology of this organ, we could see that the organoid prepared with wild-type fibroblasts developed a completely normal epidermis, while the ones prepared with NIX knockdown fibroblasts, developed epidermal layer that is very similar to squamous cell carcinoma. And we have to think that this was caused by cells that were in a different compartment. So most probably the mitochondrial defect that is happening in these cells that triggers this release of EVs containing mitochondria influences somehow in the phenotype of the skin. So this is one of the targets that we found so far. and that I would like to share with you. The second target comes from a different model. It's a model of TBHP-induced senescence. So TBHP causes oxidative stress, and we have developed this model in which we treat our cells with TBHP, similar to the UVB, twice a day for 4 days, and at the end of 14 days, we have a full senescence phenotype. And this senescence phenotype is a little bit different from the UVB-induced senescence phenotype. And by doing transcriptomic analysis, we have found that a gene that is called GDF15 was highly upregulated upon senescence induced by TBHP. GDF15, I think you might have heard about this. It's considered a cytokine, and it's considered also a novel systemic biomarker of oxidative stress, inflammation, and cellular aging. And it can have beneficial and detrimental effects in the tissue, depending on where it is acting. So since this was a target that we found regulated in our model, we decided to modulate this target. So again, we generated these GDF15 knockdown cells. And here we also found that knockdown of GDF15 induces premature senescence. But interestingly, this premature senescence of the GDF15 knockdown is characterized by a different type of mitochondrial dysfunction. In this case, our mitochondria, they are in the GDF15 knockdown cells, they are hyperfused. And we know that the fission of the mitochondria, the fragmentation of the mitochondria, it's one of the first steps for these damaged mitochondria to be eliminated by mitophagy. And in this case, we have hyperfused mitochondria, but we also have a different secretome of these cells that is characterized by upregulation of several matrix metalloproteases, but also by the secretion of extracellular particles. And here I say particles because, in contrast with the NIX knockdown cells, GDF15 knockdown cells that cannot do fission on their mitochondria, they mainly secrete cell-free mitochondria. They secrete several of these cell-free mitochondria in the extracellular environment. And what happens when we make these organoids with GDF15 knockdown fibroblasts is that the secretome of these cells modulate the skin tissue towards an aging phenotype, which means that they have decreased epidermal thickness, they have more apoptosis of the epidermal cells. And again, this is triggered by cells that are in a different compartment. So these are two different molecular mechanisms that we have found. And based on these, we found targets for intervention in the skin. And we are now trying to modulate these targets. And how do we do this? We do this together with the Institute of Pharmacy and Pharmaceutical Chemistry of our university, together with Dr. Teresa Casero. And when we find our targets, she goes back to the chemical space and she finds compounds that are already existing and are able to modulate the pathway that we are interested upon. And she works on compound optimization based on the properties of the existing compounds and the specificity of the pathway that we want to investigate. So far, we have 3 compounds that we are using for modulating GDF15 activity in the cells. And as you can see, compound 2 has a very promising activity. So it kills more than 60% of senescent cells and around 10% of control cells. And now I would like to actually tell you a little bit about issues that we have, because it's very interesting to find targets for intervention and compounds that we can use for intervening in the skin tissue. But we have 3 problems that always come to our hands when we are talking about interventions on senescent cells. The first problem is how to detect senescent cells in the skin, because so far, the main way of detecting senescent cells in the skin tissue is via biopsies. But if we think that we have to take biopsies from the elderly or people that have problems in wound healing, this is not really the best way to assess senescent cells. The second problem is that we know nowadays that senescence is not a homogeneous phenotype. So even within the same tissue, within the same cell types, senescence is very heterogeneous, and we have to be able to find biomarkers for assessing or mechanisms that we can really assess senescent cells. And third is that, as I told you at the beginning, skin is the barrier of the body. So normally, the compounds should not enter the skin. So we have to think about how to effectively reach the target, like the skin cells. So to circumvent the first and the second problems, we are working together with the Leibniz Institute in Jena for the development of non-invasive approaches to detect senescent cells in the skin. So we are using a set of imaging, different kinds of imaging, together with machine learning, such as Raman spectroscopy and microscopy, near-infrared spectroscopy, and optical photothermal infrared microscopy, among others. And so far, we are able, in 2D cultures, to completely differentiate with non-invasive approaches, completely differentiate between control and senescent cells, and also using optical photothermal infrared microscopy, we are able to differentiate between composition of proteins, lipids, and DNA between senescent and non-senescent cells. And when we are using our 3D skin equivalents, we are making them with different proportions of senescent cells, because we have to think that in the old skin, we don't have 100% of senescent cells, and the system is already able to detect and to differentiate between skin equivalents or organoids that are prepared with different proportions of senescent cells. So this is something that we are investing a lot of energy. And the other issue that I told you is how to intervene, because we have to guarantee that these compounds or this way, the modulation of the system has to reach the cells that we are interested in. In our case, it's mainly the fibroblasts we are in the dermis. So we are also investing a lot of energy on developing new approaches for skin intervention. And we are now researching therapeutic nuclear magnetic resonance that was already shown by colleagues of ours to modulate mitochondrial physiology in cells. So this would be a non-invasive methodology of manipulating mitochondrial physiology in our cells and trying to recover their function. I hope with this I could convince you about the importance of the study of cellular senescence and skin aging, to show you a little bit of our strategies to address these issues, and the efforts that we are making to find strategies for addressing senescent cells in the skin in non-invasive methodologies, and also how to target our senescent cells. And I would also like to thank my group that is very supportive in this work, and all the people that are helping directly or indirectly in this work. And thank you for your attention.
Thank you very much. Do we have any questions?
Maybe I will ask. This wonderful talk. And do you think that you can detect senescent cells, extracellular vesicles derived from skin senescent cells as a liquid biopsy in the blood of the
So in the blood,
patient?
it's very difficult, because we have to make sure about the origin of these extracellular vesicles. And so far, there is no unique biomarker to define the origin of extracellular vesicles coming from the skin. But this is a new field, and I'm sure that we and others are putting a lot of effort on developing this technology. But what we can is, so far, to isolate extracellular vesicles from the interstitial fluid of the skin. It is a very painful and long process, but it is possible.
Thank you.
I also have a question. I was wondering, what do you think happens to these extracellular vesicles carrying the mitochondria, are they delivered to other cells? Do you think they are toxic to the body?
Well, we have preliminary data that shows that they are uptaken by healthy cells. And we are now trying to investigate if they fuse with the mitochondrial network of the healthy cells, or if there they just release like a hermetic response. So this is ongoing research.
Okay, thank you very much.